Hi, my name's Andrew, and my project is titled Nonlinear Integration of Sensory Inputs in a C. elegans neuron. What do we mean by integration? For an animal, they have to integrate diverse sources of sensory information. This could be the taste of an apple or the heat from a fire, along with internal sources of information like mood or energy levels. And they often have to synthesize this information into a behavior. So when they're receiving two different sources of information, how do they combine them? On the cellular level, we're interested in two types of integration of sensory information. On the right, under E, we see that if you stimulate a neuron on two different dendritic branches, and then you record from the cell body, you receive a output that is the sum of the inputs. But on the left, when they stimulate at 2 points on the same dendritic branch, and then they record from the cell body, they receive an output that is larger than the sum of the two combined inputs. Importantly, these experiments were done in rat cortical neurons from a brain slice, not in a awake living animal. So we're interested in studying, does this superlinear summation occur in a living animal? Well, one good model organism to use is this nematode called C. elegans. It's a microscopic worm. It has 302 neurons, which develop in stereotypical ways for each animal. And we have a fully mapped connectome of these neurons. So we know how they connect to each other, and we know a lot about the promoters that lead to development of these neurons and a lot of the sensory inputs that cause activity in these neurons. C. elegans has a transparent body, so we can also image neurons as they're firing. We have a candidate neuron called AIB that we noticed had a lot of inputs clustered on one side of the neuron. This indicates it could be a good candidate for sensory integration, specifically the nonlinear type. What's good about the sensory inputs onto AIB is that we also have a way of stimulating them in a controlled setting. On the right here, we have yellow for AWC and purple for AIA. This diagram of odor response in AWC shows that AWC will reliably activate in response to an odor being presented to the worm. Now, we can also activate AIA in response to different sensory information. We want to perform experiments in which we look at how AIB responds to AIA individually, to AWC individually, and then to the combination of the two neurons. So we can provide an odor, which smells like bananas, to the worm, and this will stimulate AWC. We can also use a specific wavelength of light to activate ion channels that we have programmed to activate AIA. So AIA will respond to this 617 nanometer light. At the same time that we're activating these neurons, we can record the behavior of these neurons, specifically their voltage activity, to see how they are reacting to these inputs. We also want to try doing the light and the odor at the same time. So these are some of the tools we're using to stimulate AIA and also record from all the neurons. Channel rhodopsin on the left, it's a light-gated ion channel. So when we stimulate with this 617 nanometer light, that appears red. It's going to open up the channel in AIA, letting positive ions in, and this will activate the neuron. On the right, we have a genetically encoded calcium sensor. This is it's a GFP, which is a green fluorescent protein, and it's attached to a calmodulin protein. This means that when calcium enters the cell in response to some sort of input that's activating the cell, this will change the conformation of the protein, and it allows the green fluorescent protein to fluoresce under the 488 nanometer light. In this way, if we say that calcium influx into the cell is a proxy for neuron activity, we can also say that the green fluorescent protein, the fluorescence from this protein, is also a proxy for neuron activity. So this is what we see when we activate the neurons and we let the green fluorescent proteins fluoresce. We see that they're flickering in response to inputs. And on the right, we have activity profiles of AIB, which is the neuron that we think may involve nonlinear integration. So my project specifically is to model the activity profile of AIB and to see how the different neurons that are connected to it affect its activity. We have AVA, AIA, and AWC summed together, and each of them are weighted by alpha, beta, or gamma. So on the top, we only have these terms, alpha times AVA, beta times AIA, and gamma times AWC added together, and that is the linear version of sensory integration in AIB. If AIB is performing nonlinear integration, we need to account for that with another term, and that's where the delta times AIA times AWC term comes into play. On the bottom here, we can see that if we have no information coming from AIA, represented as a 0, but we have information from AWC as a 1, or vice versa, where AIA has information, but AWC is providing no information, we see that this term doesn't contribute to the activity of AIB. However, when we have both AIA and AWC activated, this term comes into play, and it provides that super summation activity that we saw in the rat cortical neurons. But these models are only really useful if AVA, AIA, and AWC are the main sources of activity in AIB. If there's another neuron at play, these models wouldn't be very effective. Luckily, we see that when we find the alpha, beta, and gamma weights for these models, and then we use them to reconstruct what we expect AIB to look like from the individual activity profiles, we find that we have a pretty good fit to the data. Orange is our predicted AIB activity, whereas blue is the true AIB activity. Next, to compare the linear versus the nonlinear integration models, we want to evaluate whether the quality of the fit is good. And we want to see that the nonlinear integration model has better fit than the linear integration model. This is how we calculate the residuals. We iterate through each point, and we find the difference between the expected AIB and the actual AIB trace. And then we take a square of it, and then we square root it, which essentially gives us this absolute value. When we look at the residuals for these fits, we see that on the left, we have our linear, and the far right, we have just the nonlinear terms. And both of these models give us a large amount of variance in the residuals we see for each experiment. Whereas in the middle, when we have a model with just linear and nonlinear terms, we see that the variance is a lot lower. And this tells us that that model is more consistent across worms. This experiment still has a while to go, that we need to collect more data. These values come from a set of 9 worms. We also want to make sure that the fit quality is actually as good as the residuals say they are. It's possible that adding a fourth term like AIA times AWC helps the model simply because we're adding a fourth term and not because of the nonlinear aspect of this term. We also want to make sure that when we're providing odor or light inputs, we're not activating AWC and AIA both during any of these experimental paradigms. So to do this, we're going to try to decouple the activity of the 2 neurons. We're trying to use mutants of these worms in which AWC or AIA are permanently deactivated and see how this impacts the model. I'd like to thank Amanda Ray, who's the PhD student working on this project, and my supervisor, Dr. Andrew Gordis. Thanks.